Bismillahirrahmanirrahim. Welcome to Forward Public School e-learning class 9th subject biology chapter number 5 cell cycle. I am your teacher Seema Samin. Students, today we are going to start the topic of the chapter cell cycle, that is fifth chapter. The topic is meiosis. As we have done in the cell cycle, that cell cycle is broadly divided into two phases or stages, that is interphase and division phase. We have done interphase in detail, and we also started division phase, that it further divided into two types, that is mitosis and meiosis. We have done mitosis in detail. Today we are going to discuss meiosis. For that, you must have pencil and pen in your hand to be underline the difficult words and write down their meanings. Students, for meiosis, come to page number 106 of your book. Meiosis was first discovered in 1876 by a German biologist, Oscar Hartwig. Meiosis is the type of cell division in which each daughter cell receives half the number of chromosomes as the parent cell. Meiosis is totally different from mitosis. As you remember the definition of mitosis, where mitosis is a type of cell division in which daughter cell receive same number of chromosomes as present in the parental cell. Now, if you consider this definition, meiosis is a type of cell division in which each daughter cell receives half the number of chromosomes as the parent cell. meiosis वो डिवीजन टाइप है या वो सेल डिवीजन टाइप है जिसमें कि डॉटर सेल जो है वो हाफ नंबर रिसीव करते हैं क्रोमोसोम्स का. पेरेंट सेल के जो क्रोमोसोम्स थे वो डॉटर सेल्स में हाफ हो जाते हैं. इट इज़ ऑल्सो कॉल्ड रिडक्शन डिवीजन as the diploid number of chromosomes, that is 2n, or it is also called double number of chromosomes is reduced to haploid, that is n, या फिर half number of chromosomes. ठीक है. It is also called reduction division. Reduction mean reduce हो जाना, कम हो जाना in amount, in number. So it is also called reduction division as the diploid number of chromosomes, double number of chromosomes 2n is reduced to haploid. As a result of meiosis, a single diploid cell gives rise to 4 haploid cells, as compared to mitosis, where from the one cell 2 cells produce. Here in the meiosis, from the one cell, how many cells produced? 4 haploid cells produced from one diploid cell. Meiosis is divided into two stages, that is meiosis 1 and meiosis 2. So let's go to discuss these steps one by one, that is meiosis 1 and meiosis 2. So in these steps it will be clear to you that what is diploid number, how this diploid number it is going to be reduced to haploid number or haploid cells, and how one cell going to be give rise to 4 cells. Students, as we have done on page number 106, that meiosis consists of two main stages or steps, that is meiosis 1 and meiosis 2. So let's start from meiosis 1 and come to page number 107 of your book. Meiosis 1, this phase comprises of the following stages. Like mitosis, meiosis also the karyokinases, it is also having 4 main steps or stages, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Why it have the 1? That is because meiosis, this is the first step of meiosis, that is why. Students, come to page number 107. As on the previous page we have done about meiosis, that meiosis completed or it's having 2 main step or stages, that is meiosis 1 and meiosis 2. So meiosis 1 is started from here. This phase comprises of the following stages. इस phase के, that is meiosis 1, इसके following further steps or stages है, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. So whenever a cell is going to be passed through these 4 steps, meiosis 1 is going to be completed. Then meiosis 2 is going to be started. So when meiosis 2 completed, finally one cell or parent cell gave rise to 4 daughter cells. Before going to meiosis, first or stages of meiosis, first before going to stages of meiosis 1, first come towards scientific information. In parent cell, the chromosomes are in the form of homologous pairs. This paired number of chromosomes is called diploid or 2n or double number of chromosomes. When chromosomes are not in pairs, their number is called haploid or n, a half number of chromosomes. Students, to know about diploid number, homologous chromosomes, and haploid number, just concentrate here. Just come toward this cell where it's considered parent cell, where the chromosomes are existed in the form of pair. दो क्रोमोसोम की पेयरिंग हुई है यहाँ पे. So the chromosomes which are similar in size and shape, they paired up. The chromosomes which are similar in size and shape, they paired up. And this pairing of homologous chromosome, this is called homologous formation, or simply it is called, they are called homologous chromosomes. वो क्रोमोसोम जो कि साइज़ और शेप में सिमिलर है, उनकी हो गई है पेयरिंग, और इस पेयरिंग को कहा जाता है होमोलॉग फॉर्मेशन, और सिंपली ये क्रोमोसोम एक दूसरे के होमोलॉग है. और इस स्टेज पे इसको कहा जाता है, इस पेयर को या इस पेयर को, दैट दे आर डिप्लॉयड नंबर. यहाँ क्रोमोसोम का नंबर क्या है? डिप्लॉयड. सो द क्रोमोसोम्स विच आर सिमिलर इन साइज एंड शेप, दे आर कॉल्ड होमोलोगस क्रोमोसोम्स. अब आप इस सेल के कंपैरिजन में यहाँ देखें, इन दिस वन सेल द पेयर is now like here a single chromosome existed. In this one pair also single chromosome existed. So the chromosome number gets haploid here, or it's get haploid or half. So this is the difference between diploid and haploid, and this is homologous chromosome. You must be familiar with this term. यानी कि homologous chromosome कौन से होते हैं? Which are similar in size and shape. एंड जब ये क्रोमोसोम पेयरिंग करे तो पेरेंटल सेल में इनको क्या कहा जाता है? डिप्लॉयड नंबर. बट वेन एवर अ सेल इज पास थ्रू मायोसिस, सो दिस डिप्लॉयड नंबर गेट्स रिड्यूस टू और गेट्स डिक्रीज टू हाफ नंबर, जो कि आपको यह नजर आ रहा है. एंड नाउ इट इज कॉल्ड हैप्लॉयड नंबर. अब इसको क्या कहा जाएगा? हैप्लॉयड नंबर. So in meiosis 1, the diploid homologous chromosomes in parent cell separate to produce haploid sets of chromosome in the daughter cell. जब सेल meiosis 1 से पास होगा तो जो डिप्लॉयड होमोलोगस क्रोमोसोम्स पेरेंट सेल में मौजूद हैं वो सेपरेट हो जाएंगे और उनसे क्या बन जाएगा? हैप्लॉयड सेट ऑफ क्रोमोसोम्स इन द डॉटर सेल. इस फॉर्म में जो पेयर की फॉर्म में क्रोमोसोम्स मौजूद होंगे और जब सेल पास होगा थ्रू meiosis 1 तो क्या हो जाएगा? इन द सेल द क्रोमोसोम नंबर विल बी रिमेन और विल बी एग्जिस्टेड इन सच वे. नाउ इस फॉर्म में आ जाएगा जैसे एक सेल एंड सपोज अनदर सेल. ठीक है. सो द डबल नंबर नाउ डिस्ट्रीब्यूटेड इक्वली ऑन टू सेल्स, each cell receiving one chromosome. In meiosis 1, the diploid homologous chromosomes in parent cell separate to produce haploid sets of chromosomes in daughter cells. Students, now come toward prophase 1. Prophase 1 usually accounts for 90% of the total time spent in meiosis. meiosis में जो टोटल सपोज 1 आवर लगता है, तो सो इन दिस 1 आवर 50 मिनट्स सपोज टेकन बाय prophase 1, and the remaining process, that is metaphase 1, anaphase 1, telophase 1 and meiosis 2, that is completed in this 10 minutes suppose. And if suppose we consider its percentage wise, so 90% time is taken by prophase 1 and remaining 10% time is that is enough for the rest of meiosis. In this phase, the chromatin material condenses, that is thicken, or they become thicken, and chromosomes become visible, that is clear. However, unlike prophase of mitosis, the two homologous chromosomes, the chromosomes similar in size and shape, or in shape and size, homologous chromosomes similar in size and shape pair up or synapse to form tetrad, because a pair of chromosomes have four chromatid. This one structure, it is it is called synapse, or here they are called tetrad because one, two, three, four chromatid are existed here. So this four chromatid structure it is called tetrads. ठीक है, यानी कि जैसे कि हमने प्रोफेज ऑफ माइटोसिस पढ़ा था, वहाँ ऐसा कोई पेयरिंग सिस्टम हमने नहीं देखा, बट प्रोफेज ऑफ मायोसिस वन या प्रोफेज वन, इसमें क्या हो जाएगा, द क्रोमोसोम्स विच आर सिमिलर इन साइज एंड शेप दे विल बी गेट्स पेयर अप, इनकी क्या हो जाएगी, पेयरिंग हो जाएगी, ये पेयर बना लेंगे। तो इस तरह अगर दो यानी सेम साइज और शेप के क्रोमोसोम पेयरिंग करते हैं तो हमारे पास एक पेयर में कितने chromatids आ जाते हैं, फोर chromatids. So that is why they are called tetrads, or the structure which is formed by the two similar chromosome, it is called homologous structure, or simply they are called homologs of each other. ये एक दूसरे के क्या है, होमोलॉग है। देखें आप, homologous chromosomes condense, homologous chromosome condense, that is they become thick. अब आप देखो सिस्टर क्रोमैटिड्स, द टू क्रोमैटिड्स जो कि एक क्रोमोसोम के आर्म्स हैं वो एक दूसरे के सिस्टर है। अब आप देखो, दीज टू आर सिस्टर, द ब्लू वन एंड द रेड वन आर सिस्टर, बट द वन रेड एंड वन ब्लू दे आर नॉट सिस्टर ऑफ ईच अदर। ठीक है, सो the two non-sister chromatids of homologous chromosomes become zipped together, forming complexes known as chiasmata, singular chiasma. अब आप देखो यहाँ पे भी, यहाँ पे जस्ट ये करीब आए हैं और यहाँ पे इनके दरमियान क्या प्रोटीन स्ट्रक्चर इज गोइंग टू बी प्रोड्यूस, दैट इज synaptonymal complex forming, that is synapsis begins, synapsis pairing, synapsis का आप लिखे pairing of homologous chromosome, pairing of homologous chromosomes। आप देखो पहले homologous chromosome करीब आए हैं और यहाँ पे इनकी हो गई है जॉइनिंग स्टार्ट या पेयरिंग स्टार्ट हो गई है, जिसको कहा जाता है साइनेप्सिस बिगिंस या साइनेप्सिस स्टार्टेड हेयर, पेयरिंग स्टार्टेड हेयर। अब आप देखो हेयर वी हैव सम नेटवर्क लाइक स्ट्रक्चर, एक नेटवर्क की तरह का स्ट्रक्चर प्रोड्यूस हो रहा है यहाँ पे, विच इज़ कॉल्ड साइनेप्टोनिमल कम्प्लेक्स फॉर्मिंग। इन होमोलोगस क्रोमोसोम में साइनेप्सिस जब हुई तो क्या चीज़, कौन सा स्ट्रक्चर डेवलप हुआ, साइनेप्टोनिमल कम्प्लेक्स फॉर्मिंग। सो राइट डाउन अबाउट साइनेप्टोनिमल कम्प्लेक्स, a protein structure that forms between two homologous chromosomes during meiosis 1, which helps in crossing over and chromosomal recombination. Homologous chromosome के दरमियान एक प्रोटीनेशियस या प्रोटीन containing और protein structure is produced, and this structure is important forward to be help in crossing over, that is the exchange of segments between the similar chromosomes। ठीक है, सो आप देखो फर्स्ट स्टेप पे हुआ हमारे पास होमोलोगस क्रोमोसोम्स कंडेंस, दैट इज दे बिकम थिक। सेकेंड स्टेप पे आप देखें बिटवीन दिस होमोलोगस क्रोमोसोम व्हाट हैपेंड, साइनेप्सिस बिगिंस, इनमें क्या चीज़ शुरू हो गई है, साइनेप्सिस। और थर्ड स्टेप पे आप देखो बाइवेलेंट्स फॉर्म्स, अब इनमें क्या बन गए हैं, बाइवेलेंट मीन पेयर ऑफ होमोलोगस क्रोमोसोम, एक पेयर को भी हम बाइवेलेंट कहेंगे, दूसरे यानी जितने भी क्रोमोसोम हैं दो क्रोमोसोम कंबाइन हुए जो कि एक जैसे थे और इनकी क्या बन गई है, इनकी पेयरिंग हो गई है, और इस पेयर को इस स्टेज पे क्या कहा जाता है, बाइवेलेंट। इस डिफरेंस को आप समझे, बाइवेलेंट मीन पेयर ऑफ होमोलोगस क्रोमोसोम, बाइवेलेंट पेयर ऑफ होमोलोगस क्रोमोसोम। एंड व्हाट अबाउट टेट्राड मीन whenever the four arms we consider, जब हम 1 2 3 4 या 1 2 3 4 4 आर्म्स को ले, सो दे आर, दिस स्ट्रक्चर इज कॉल्ड टेट्राड, दिस वन ऑल्सो कॉल्ड टेट्राड, एंड इट इज ऑल्सो कॉल्ड बाइवेलेंट। टू क्रोमोसोम के बेस पे इसको बाइवेलेंट कहा जाता है, एंड फोर क्रोमैटिड के बेस पे इसको कहा जाता है टेट्राड। अब आप देखो दीज टू रेड वन आर सिस्टर ऑफ ईच अदर, they are called sister chromatid, and the blue one are sister of each other, that is they are sister chromatids, but the red one and the blue one they are not sister, they are called non sister chromatids। जैसे कि 1 और 2 sister chromatid है और 3 और 4 sister chromatid है, but 2 and 3 are not sister chromatid, they are called non sister chromatids। Clear? At chiasma, this one structure, that is chiasma. At chiasma, the non-sister chromatids of homologous chromosome exchange their parts. At chiasma, the non-sister chromatid, the 2 and 3, they will be exchanged their parts. Just come toward this one structure where the red chromatid gets a part of blue one and the blue one gets a part of red one. So at chiasma, the non-sister chromatids of homologous chromosomes exchange their parts. They exchange their parts. This process is known as crossing over. आप देखो at 4 step what happened? Crossing over occurs. First chromosome condense, second pairing begins, third this pairing completed, and at the fourth step they they exchange their segment with each other, the non-sister chromatid. For example, 2 and 3, they exchange the segment, not 1 and 2, not 3 and 4. Exchange is not done between 3 and 4 and 1 and 2. Exchange is done between non-sister chromatids, that is between 2 and 3, that is between red and blue. At chiasma, the non-sister chromatids of homologous chromosomes exchange their parts. This process is known as crossing over. As in the prophase of mitosis, here also, students, after crossing over, come to this one diagram. The chiasma becomes visible as the chiasma, this one structure become visible, which is simply the exchange of genetic material between the non-sister chromatids. आप देखें, the blue one chromosome having a part of red chromosome and red chromosome having a part of blue chromosome. So the chiasma becomes visible as chromosome arms separate. क्रोमोसोम आर्म सेपरेट, क्रोमोसोम आर्म एक दूसरे से क्या हो गए? सेपरेट हुए, मूव अपार्ट, थोड़े से अलग हो गए हैं, यानी थोड़ा सा दूर हो गए हैं. आर्म सेपरेट ड्यूरिंग लेट प्रोफेज, यानी प्रोफेज के एंड पे, जब प्रोफेज एंड होने जा रहा हो, द क्रोमोसोम बिकम मच मोर थिक एंड देयर पार्ट्स गेट अ बिट सेपरेटेड बट स्टिल दे आर अटैच्ड एट सेंट्रोमेयर. ठीक है. So in the prophase of mitosis, here like the same like first the most important process which is take place here, that is crossing over. After the crossing over, the as in the prophase of mitosis, here also the nucleoli disappear. The nucleoli, the structure, the rounded structure which are present in the nucleus, they disappear, they dissolve, they get disperse in the cytoplasm, and the nuclear envelope breaks, and the envelope which is existed around the nucleus, it is also broken down. Centrioles migrate or move to opposite poles and make spindle fiber. So these steps, that is the nuclear envelope disappearance, that is nucleoli disappearance, and the centriole formation, that is spindle fiber formation, these all steps, they are same like prophase of mitosis. Students, here in this diagram we have these 4 events of meiosis 1, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. As on the previous page, that is 107, we have done about the most important events which are taking place during prophase 1. So they are concluded here, or it simply started from here. यहाँ आप देखें, वहाँ जितनी बातें हमने देख ली वो सारा आपको यहाँ सेल के अंदर बता रहे थे. दैट फॉर योर कन्वीनियंस, वहाँ पे क्रोमोसोम को सेल से निकाल के आपको बताया जा रहा था. यहाँ पे वही सारा प्रोसेस सेल के अंदर आप देखो. चियाजमेटा मीन chiasma, that is simply the crossing over, the exchange of segments. Chiasmata is plural term, chiasma is a singular term. जहाँ पे होमोलोगस क्रोमोसोम में क्रॉसिंग ओवर एक से ज़्यादा एरियाज या पार्ट्स में हो, तो ऐसे में इसको क्या कहा जाता है? चियाजमेटा. आप देखो, सेंट्रोसोम, दैट इज सेंट्रोसोम इज स्ट्रक्चर फॉर्म बाय द सेंट्रियोल प्लस स्पिंडल फाइबर. स्पिंडल फाइबर और सेंट्रियोल जब कंबाइन हो जाते हैं, या फिर जहाँ से ये सेंट्रियोल से स्पिंडल फाइबर अराइज होते हैं, इस एरिया को क्या कहा जाता है? सेंट्रोसोम. न्यूक्लियर एनवेलप फ्रेगमेंट्स, fragment mean parts. Nuclear envelopes going to be disappear or it's going to be breaks down. आहिस्ता आहिस्ता न्यूक्लियर एनवेलप क्या होना शुरू हो जाती है ड्यूरिंग प्रोफेज? इट इज डिवाइडेड इनटू पीसेस और पार्ट्स, मेकिंग और लेटिंग द क्रोमोसोम टू बी गेट्स फ्री in the cytoplasm. Homologous chromosomes आपको बताए जा रहे हैं. The chromosomes condense, same, they become thick, and the nuclear envelope breaks down. और nuclear envelope क्या हुई? इसकी हो गई, यानी कि ये ख़त्म हो गया है. And crossing over occurs. So the most important events which occur during the prophase 1 were the chromosome condenses and the nuclear envelope breaks down and the crossing over, that is exchange of segments between the similar or the homologous chromosome, is done during the prophase 1. Now cell is ready to be entered in the next subphase of meiosis 1, that is metaphase 1. During metaphase 1, what happened to the chromosomes? During metaphase 1, the chromosomes gets arranged themselves in the center of the cell, making metaphase plate, as like the mitosis. So look here, that is metaphase 1. Homologous chromosomes arrange themselves during the metaphase plate. During the metaphase plate, what happened? The homologous chromosomes arrange themselves. But here is a difference between mitosis and meiosis. If you remember mitosis, there was single chromosome arranged in the center. But here, chromosomes arrangement take place during metaphase 1 in the form of pairs. आप देखो, हर एक क्रोमोसोम जो है वो सिंगल नहीं है. ड्यूरिंग मेटाफेज, जैसे यहाँ पे क्रोमोसोम होमोलोग फॉर्म में एग्जिस्ट कर रहे थे, द सेम होमोलोगस पेयर गेट्स arranged in the center of the cell now, making a plate-like structure which is called metaphase plate. So, homologous chromosomes arrange themselves during metaphase plate. Spindle fibers from one pole attached to one chromosome of each homologous pair, while the spindle fiber from the opposite pole attached to the other chromosome of the homologous pair. आप देखो, if we consider one homologous pair, suppose red and blue chromosome, so the red chromosome will be attached by a spindle fiber from the one pole, and the blue one will be attached by the spindle fiber from the opposite pole. Similarly, here the red one will be attached or will be arranged from the this one respective pole and the blue one from the lower pole. So in such way, each and every pair, it will be having spindle fiber in this spindle fiber, that is one member will be attached from one pole and the other member will be attached from the opposite pole. Now come towards anaphase, that is anaphase 1. Now what happened during anaphase 1? During anaphase 1, the spindle fiber pull towards the poles. The spindle fiber will be pulled towards the poles. Spindle fiber कहाँ जाएंगे? They will be moved toward the poles. So each member of the pair is pulled towards the opposite poles of the cell. Look here to this one pair. The red chromosome is pulled toward one pole. The blue one is pulled toward the other pole. Similarly, in this pair also, one chromosome pulled toward one pole, the other chromosome pulled toward the opposite pole. In this pair also, one chromosome is pulled toward one pole and the other chromosome is pulled toward the other pole, making the center free. Center क्या हो रहा है? Slowly and gradually it gets free and chromosome is now pulling toward the poles. During this phase, the sister chromatids remain attached. During this phase, if we consider this chromosome, so the sister chromosome, they remain attached. So chromosome move as a whole. Here also the chromosome move as a whole to the opposite pole. So the sister chromatids like this one and this one, they are sister. They remain attached. In this way, one haploid set of chromosome is formed at each pole. First it was double, that was in the form of pair, but now at each pole it is existing now in the form of single chromosome. Like one chromosome will be moved to one pole, the other will be moved to opposite pole. Similarly here, one chromosome will be moved to one pole, the other will be moved to the opposite. Same happened to this pair also. So in this way, one haploid set of chromosome is formed at each pole. Each chromosome in both haploid set still contain two sister chromatids. आप देखो हर क्रोमोसोम, यहाँ पे भी अगर तीनों देखें और यहाँ पे भी, each chromosome consisting of a sister क्रोमैटिड्स, हर क्रोमोसोम जो है इस तरह इस फॉर्म में नहीं गया, बल्कि इस फॉर्म में गया है, वेयर क्रोमोसोम इज नॉट एग्जिस्टेड इन अ सिंगल वे, इट इज एग्जिस्टेड इन द फॉर्म ऑफ टू सिस्टर क्रोमैटिड्स। इस डिफरेंस को आप समझे। माइटोसिस पे इस फॉर्म में क्रोमोसोम जब पोल पे पहुंचे तो ऐसे थे, बट यहाँ पे whenever they move toward the poles, they are existed in the form of in this way, that is they having sister chromatids, they are double. Now come towards telophase 1. After the chromosome reach their respective poles, the nuclear membrane reappear along the appearance of the nucleoli. The nucleoli and the nuclear membrane, which was existed in a dispersed way in the cytoplasm, now again they start gathering at the poles. So in such way, they gathered around the chromosomes on the poles. Later, cytokinesis occurs and the two haploid daughter cells are produced. Cytokinesis, that is simply division of cytoplasm, take place, and now from the one cell, how many cells produce? Two cells produce in the telophase, that is telophase and cytokinesis. Chromosomes gathered at the poles of the cell. Chromosome gathered at the poles of the cells, the cytoplasm divides. The cytoplasm divides. So like it's considered animal cell, that is why it is divided by cleavage furrow, that is the cell membrane gets deepened from the center and finally divide one cell into two. Now if you consider from the start, that is prophase 1, and comes to telophase 1, so here in the parental cell there were 6 chromosomes, but now here at the end of meiosis 1, in the two cells, each cell having 3 chromosomes, that is 1 2 3 1 2 3. Here we have 6 chromosomes. And if you consider this diagram, here the chromosomes are not exactly like the parental cell. They have an exchange segments. If you consider this blue chromosome, it having a part of red chromosome. Similarly, the red chromosome having a part of blue chromosome. Here also, the red chromosome have a part of blue chromosome and the blue one having a part of red chromosome. Now come towards scientific information. Anaphase 1 is a key event in creating new genetic combination in sexually reproducing organisms. Like this process, the whole process of meiosis is dependent on anaphase 1 because it results in such cells where the chromosomes are recombined. They are not existed exactly like the parental chromosome now, they have exchange segments in them. So, new combination of genetic material created by any process, including crossing over, is termed as genetic recombination because this cell is passed from crossing over. So that is why these chromosomes they are now genetically like they have changed genetic makeup whenever they will be participate in fertilization, so the organism which will be produced as a result of these, they will be having variations. So in such way, crossing over as well as anaphase 1, they are considered the basis of variation in the living organisms. Students, this was the end of meiosis 1. Now we are going to start meiosis 2, where these two cells will be now considered or they will be participate in the further step and will be going to be give rise to 4 cells. That is simply in the first step from the one cell 2 cells produce, and in the meiosis 2 from the 2 cells 4 cells will be finally produced. Students, this was the end of meiosis 1. Meiosis 2 will be discussed in the next class, that is topic number 4. Thank you.